Hello everyone, my name is Shiraz Mumtaz. Today we're going to learn about the neuroglial cells. The objective is basically the understanding that what are those neuroglial cells, which means that what types of those neuroglial cells, and also what are their functions. Okay. So to begin with, the neuroglial cells actually it is the cells that they support and protect the neurons. Okay, I mean we have the structure by itself, like which is the neuron by itself. Okay, and the neuron by itself, okay, like which is like the axon, the neurocell bodies and everything, the dendrites coming out, the axons are coming out, okay, but we need somebody to protect it, okay. So there are different types of cells that protect it, like there are the astrocytes, the ependymal cells, the oligodendrocytes, and the microglia, okay. So basically, these neuroglial cells make up to half of the volume of the nervous system. Okay, so including everything, like when you see like four of this is contained almost like 50%. Okay, the rest of it is going to be the neuron, like which process the information. Okay. All right, so that's what it looks like. Okay, the picture like the ependymal cells, okay, that secretes actually the CSF, have the what's it called the cilia that helps in like circulation of the CSF, which we're gonna come in a minute. Okay, we're gonna go through everything just to show you how those microscopically look like those microglial cells. Okay, we are gonna discuss about much of a what is it called the satellite cells and the Schwann cells, because they're basically they're neuroglia, but not for the central nervous system, which is the brain or the spinal cord. Okay. All right. So neuroglial cells or astrocytes. Okay. So first is the astrocyte. Okay. It is the most abundant cell which is available in the CNS. The basic function of this astrocytes is maintain the blood-brain barrier. Okay, blood-brain barrier is like actually a selective barrier that isolates the brain from the general circulation. So which means that like whatever we have in our circulation, general circulation, even like any infection or even glucose or whatever, it is different than whatever we have in our central nervous system. Okay, so composition is totally different. Okay, if you consider like the blood glucose and everything, you're gonna find a different thing. You're gonna find the not very much of the white blood cell, which is the microglia, which is the fixed macrophages. Okay, which we're gonna come in next. Okay, we're gonna do in detail. But it also gives the astrocyte gives a three-dimensional shape to the brain, the three-dimensional shape to the CNS. Okay, it repairs the damage of the nervous system, but not exactly, not the neuron. Okay, so a neuron by itself. Okay, when you see that like the neuron, I mean I'm always the making those weird kind of a neuron, okay, but it's just like telling you like this is the dendrites coming out and there's an axon coming out. So basically the astrocyte does not regenerate the neuron by itself, but it can regenerate the dendrite, it can regenerate the axon and axon helix and all that, or the collateral if coming out from that axon. So there's example. Okay, um, so astrocytes also controls and regulate the concentration of the iron, okay, nutrition and waste, and recycle the neurotransmitters, which is the mainly, okay, so neurotransmitters which helps in like sending the information from one side to another side, okay, it recycles those, okay, and helps in communication with the other neurons as well, okay. Now comes the oligodendrocytes. The oligodendrocytes, okay, it is a kind of a cover for your cord, okay. Whatever you see, like cord around, like there is a the metal or copper wire inside, most commonly, okay, and there you see a plastic cover on the top, so that none of the electric current loose around it. So same thing we have in our body, okay, that we have the oligodendrocyte, then makes up that sheath, okay, then makes up the sheath. This is a nervous tissue right here. This is a nervous tissue right here around the sheath. Okay, that helps in actually increasing the propagation from one side to another side. Okay, going towards more away. Okay, whatever interior is going there. So it increased the speed faster from the action, like from one place, traveling from one place to another place. Okay. All right, so this is the oligodendrocytes, which is the making of the myelin sheath within the central nervous system, only the central nervous system, which is the brain or the spinal cord, not anywhere else, not the peripheral nervous system. Okay. All right. So right here, the myelin is what is it called? A lipid insulation that increases the speed of propagation of action potential that I was talking earlier. Okay. Then comes the internode. Internode is this thing which is the myelin sheath, and we have a node of Ranvier. Okay, so right here we do not have a what is it called, myelin sheath. Okay, and then actually the what is it called, the action potential like within it go from one place, like you're traveling from jumping side, like from one side to another side and then to another side to another side, unlike the unmyelinated, which is does not have a jumping pattern. So if it does not have a jumping pattern, it goes slow. This one is faster. Okay, and faster means like that if you have the myelinated sheep, like myelinated axons, so which means that like it used for the threatening condition, like if you want to run away, like some dog or lion or tiger, God forbid, is following you, so you're gonna use this kind of a axon, which are the myelinated. Okay. And this actually, the oligodendrocyte, makes the central nervous system appear white. If you ever seen like the cerebral cortex, like which is cerebellum, the biggest part. So right here we see like if you see the cross section around it, okay, you can see the coronal section. So right here we see different kind of like the gray matter is in outside, and then white matter is inside. So wherever you see a white matter inside, okay, which is the more of the myelinated, right here the gray matter is outside in the cerebellum, and then white matter is inside. So what is inside, it has the more of the white matter, which means it has the myelin sheath. Okay. All right. Now, then come the ependymal cells. Ependymal cells, okay. Actually, these are the ventricle system, okay, that within the we call the central nervous system and goes through all the way toward the spinal cord, okay. So different kind of ventricles, like that we have, and ventricle system is lined by a structure called a choroid plexus. Choroid plexus is actually lined by a cell called a ependymal cells. These are the ependymal cells, as you can see right here. And then this is actually a CSF inside, and then there should be a blood vessel outside. Okay, so blood vessel, like send the the limited amount of like fluid inside, and then it can cross. Okay, and then goes inside. And this one right here, these are the cilia that helps in the regulation of the CSF within the subarachnoid space that is there for the cerebrospinal fluid. Okay, so cilia helps in the circulation, and then there are the ependymal cell, which is helps in what is it called, the what is it called, transportation, kind of a blood, not 100% blood, that goes into and take the nutrition, okay, to the within the cerebrospinal fluid. Okay. All right, now comes the microglia, not the last, but also the least, because the thing is that the microglia, they are the fixed macrophages in different structures, we have the fixed macrophages. For the central nervous system, we have the fixed macrophages that remains in there. It does not go out at all from and see the blood at all. Okay. So this is the least amount of least numerous neuroglia, because the thing is that we have, as we discussed earlier, we have the astrocytes that maintain the blood-brain barrier. If you have a good what is it called, barrier between the circulation, the normal circulation, and the brain, then you don't need much of a microglia. Okay. But this microglia actually helps in what we call whatever we have the cellular debris or waste product, and remove the pathogen by phagocytosis. That is the function of the all the macrophages. Okay, so all the macrophages have the function to phagocytose and digest and dissociate, get rid of the now we call the pathogens. So that's what the function of the microglia. And as we discussed, that this is the very least amount of neuroglial cell, because we have the strong barrier, so none of the infection can cross from the general circulation and pass through the CSF. Okay. Anyways, thank you very much. Appreciate your time.